So let's talk about a functional protein, okay, in enzymes. Enzymes are proteins that act as biological catalysts. On the surface of the enzyme, a small region that's called the active site binds to a substrate and it catalyzes a specific reaction for that substrate. So what does that mean? The active site is kind of like the front door. Okay, the substrate, that's a fancy word for starting material. Catalyzing, the definition of catalyzing is that it makes enzymes make things quicker and easier by catalyzing that reaction. Okay, speeding it up, catalyzing, speeding it up. So enzymes are biologic catalysts. So they catalyze nearly all chemical reactions by taking place that are taking place in the cells of the body. They increase the rate of reaction by lowering the energy that's required. And the enzyme carbonic anhydrase lowers the activation energy for the reaction, specifically for this one. Okay, so if the blue line here is the activation energy without an enzyme, and the purple is activation energy with an enzyme, you can see that it takes less energy to get the reaction done. So I like to say on a normal day, it takes me 5 cups of coffee to get out of bed, but an enzyme would make it 2. So it's quicker and easier for me to get out of bed. Okay, so how do we name them? Well, it's derived by placing the end of the name of the reaction or the reacting compound by the suffix "ase." So it identifies the reacting substance. For example, sucrase is going to be catalyzing or doing the reaction of sucrose. So you have sucrose here, and then we end it in "ase," and that's what makes it the enzyme. Okay, it also can describe the compound or the reaction that's catalyzed. For example, oxidase is going to catalyze an oxidation reaction. And it also could be a common name, particularly for the digestion of enzymes such as pepsin and trypsin. Okay. So there are a couple different classes, and enzymes are classified by the reactions that they catalyze. And there are 6 main classes of enzymes. Okay, so you have oxidoreductases, and what do? Redox reactions, so oxidation reduction. Transferases are going to transfer groups of atoms. Hydrolases are going to be, we're using water to break things apart, hydrolysis. Lysases, these are adding or removing atoms to form a double bond. Isomerases, remember the isomer is different rearrangements of atoms, so the isomerases are going to rearrange atoms, where ligases, to ligate means to glue together, okay? And so you're going to use the ATP to combine the small molecules or ligate them together. So let's match the type of reaction catalyzed with the enzyme class. Pause me and come back. All right, so when we have forming bonds, we're gluing those together, that would be the ligase. Rearranging atoms, that's making the isomer, so isomerase. Transferring a group, transferase, because transfer. And then hydrolysis is the hydrolases. Okay, great job. All right, so how do these actually work? Well, using active sites to bind the substrates. Okay, this is how proteins actually work, or these enzymes actually work. On the surface of an enzyme, a small region that's called an active site that is going to bind to the substrate, which is a fancy word for starting material, and catalyze or do a reaction of that substance or that substrate. Okay, so the active site is the region where the enzyme fits the shape of the reacting molecule, and that's called the substrate. And then this contains the R group, the amino acid R groups that bind to the substrate, and then it releases the products when the reaction is complete. So they're very specific. The active site is very specific for shape, okay, because it can only bind to one type of amino acid or enzyme, substrate to an enzyme. All right, so in an enzyme-catalyzed reaction, the substrate attaches to the active site, and then that becomes the enzyme substrate complex. Complex meaning 2 things together. Then this reaction occurs, and the products are made, and it becomes the enzyme product complex, and then we release the product. And then the enzyme gets to be used over and over and over. Okay, so we have our enzyme and substrate. It binds together to make the enzyme substrate complex, and it does the little reaction, and then the enzyme is going to be recycled to be used again, and it's going to be releasing the product. Okay, and you can see the same thing here. So binding of a substrate occurs when it interacts with the amino acids of the active site. So with enzyme-catalyzed reactions, in the hydrolysis of the disaccharide sucrose, di meaning 2, saccharide meaning sugar, so the name of that would be sucrose. And remember, hydrolysis is using water to lyse or break apart. Okay, so then the enzyme substrate complex is formed as sucrose binds to the active site of sucrase. Okay, remember ACE is the enzyme. And then the glycosidic bond of sucrose is in the position for hydrolysis, using water to break apart that bond. Then the R groups of the amino acids in the active site catalyze the hydrolysis of sucrose, producing glucose and fructose, the two constituents of the sucrose. And then the product structures are no longer attracted to the active site, so they are released to allow sucrase to interact with another sucrose molecule. That was a mouthful, but basically you have your starting enzyme sucrase plus the substrate sucrose. Combine them together, it makes the enzyme, or the sucrase the enzyme substrate complex. Then it gets turned into the product of sucrase and the enzyme being recycled. I'm sorry, sucrase, and then the product is being released. Okay, so when we talk about enzyme reactions, we call this a lock and key model. And so in the lock and key model, the active site has a rigid, non-flexible shape, and the enzyme binds only to the substrates that fit exactly into the active site, like a lock. And then the substrate is the key that fits into that lock. And this model was a static one that did not include for flexibility of the tertiary shape of the enzyme and the way that the active site can adjust to the shape of the substrate. So then we have the induced fit molecule model that came into play. So the enzyme structure is flexible, not rigid, and it adjusts to the shape of the active site in order to bind to the substance. So this is the range of substrate specificity, and it can increase because of this, and then the shape changes improve the catalytic reaction during the reaction, catalysis during the reaction. Okay, so basically, instead of just being completely rigid and be like, oh, you fit, great. Induced fit, I say it's like a little snuggle. So once that substrate binds to the enzyme kind of wraps around it and gives it a little snuggle, and that would be the induced fit. So again, in the induced fit model, the substrate and enzyme work together to acquire a geometric arrangement that lowers the activation energy of the reaction. So again, remember, just enzymes make things quicker and easier. Okay. Yeah, I'm going to stop right there.